So what we're going to do is a Q&A. So this is your chance, and no question is a stupid question. So, and yes, get in line, get up here. You can ask questions, and then I know folks listening online, you could type your questions in. We're moderating that as well, and we'll get going. Is that working?
Hello. So you said you were doing newborn mice and then a 1-month-old mouse. So that's amazing because that's like adult, right? 1 month. So what is the equivalent age in a person for 1 month or whatever?
So not surprisingly, the mice age is not exactly translated human age. It's like you're trying to figure out how old my dogs are in human age. But generally, P30 mice are considered adolescent stage, that they're maturing, but not quite adult. Yeah, that's about the stage. And now we are testing that in adult mice. So about 60 days old mice are considered fully grown adult mice.
Hello?
My first question is whether the mice models have any other phenotypic or behavioral outcomes that you're tracking, like seizure frequency, and whether you're rescuing the mice from having seizures with the gene therapy?
So the mice model have the behavior phenotypes that they move less and they have repetitive movement, so they're like self-grooming a lot. They also show from time to time spontaneous seizures, and our collaborating lab showed that when they measure the EEG, they have human FOXG1 patient-like spikes or spike slowing process. And now whether we can recover that in mouse model, that's what we are testing. So we tested the ultrasonic vocalization as a proxy for the behavior communication phenotypes, and testing this behavior, other behavior that I mentioned, requires their growth to adult. So we did the main test in P30, which is to adolescent. So we'll use this behavior platform that we developed for testing that in behavior levels.
And my other question was, of the different sort of pieces that you mentioned, the oligodendrocytes and the microglia, do you have a sense for if any one of those is more important to like function to rescue than another, if it, say, as an addition to the gene therapy for one of these small molecules, if we're targeting one or the other?
Right. So we haven't really teased out which one is the major contributor of the FOXG1 syndrome symptoms. So that's what we are doing. We are deleting FOXG1 only in oligodendrocyte, for instance, to see whether the mice show the FOXG1 syndrome-like symptoms. We know that immune activation is indirect from indirect because FOXG1 is not expressed in the microglia cells. What I can tell you is I think that all these symptoms or all these disease mechanisms will work together to make a full-blown FOXG1 syndrome symptoms. And if we divide them into small pieces, then that might lead to less severe symptoms. But that needs to be tested. One thing that really going for our way is that we need to increase FOXG1 levels in neurons. We don't have to add FOXG1 into oligodendrocyte or microglia. If we delete the FOXG1 only neurons, they have full-blown symptoms. If we put the FOXG1 only two neurons back onto these mutant mice, then it shows a lot of recoveries as well.
We have a question from the virtual attendees, which we have over 50 from around the world. I see Slovenia and Italy, and thank you so much for attending virtually. This one's for Guy. It says, often the bottleneck in rare disease pharma partners because there is no money in curing rare diseases, how do we handle that?
Okay, how do we handle it? We do it ourselves. That's how we handle it. We basically grab the bull by the horn and we drive this not just into clinical trials, but through clinical trials. We're not here to replace pharma. I think that at the end of the day, partnerships with pharma are going to be important and actually essential. I think that the key is timing. And what we're doing is we're taking on ourselves the advancement of the program through clinical proof of concept. In other words, we will be running the phase 1, phase 2, and demonstrating, hopefully, a robust treatment effect. At that point, if we partner a program that demonstrated clinical efficacy. That makes it a lot less likely that a pharma partner will drop it. Right? Pharma partners drop programs based on portfolio prioritization when the risk is high, and then they need to prioritize. We basically take on the risk in terms of, I'm talking about the monetary risk, right? By taking on the cost and the work through clinical proof of concept, we are de-risking the program from a business perspective. So we're not here to replace pharma, but we are here to take it through the stages where most pharma partners are less interested because the business risk is high.
And Ryan, before you go on, if anyone has a question in the room, just stand up. So, okay, I think there's one back there.
We have
Dr. Benke.
one more quick one. I think it'll be very quick from the virtual attendees, which is, are there other viral-based gene therapies curing rare diseases now?
Other virals, I'm not sure if other AAVs, so not just AAV9 or...
Yeah, so
I mean, we
like
know
Zolgensma and these other viral-based
So
gene
there
therapies.
are AAV treatments in the clinic, and those are, showing in what I would regard as very encouraging clinical data, right? And we are all looking forward to see how those will move through the clinic and whether they'll get approval. But I think that across the board, whether it's AAV or antisense oligonucleotides for other conditions, for example, Angelman syndrome, there is very encouraging emerging data to suggest that patients are improving on treatment.
Turn it off and on, see if that works. Yeah. Hi, Tim Benke. I'm medical director at the Rat Clinic at University of Colorado, Children's Hospital, Colorado. So there are 2 clinical trials for neurodevelopmental disorders using gene replacement therapy for Rett syndrome. And we're very early on in those. And we can talk offline, but there's been a lot going on in that trial over the last week. And it has a lot to do with safety. So I was wondering if you could address how you're going to measure for toxicity in your mouse models, because we know that too much FOXG1 is a thing. So how are we going to measure? So my thought is find what that point is. You need to have something that tells you, okay, I've now put in too much. And so we need to know where that line is for too much. And I'm wondering if you can tell me what that looks like. What does too much look like in your mouse model?
So this is a super important question, and one that we are addressing. We're addressing in 2 different ways. One of them at UB with a study that is already ongoing in Soo's lab, and the other is through safety studies that we're doing in other species with Charles River. And so just to contextualize, there's you want the right amount of FOXG1 protein. We know that too little is not good, but is there a risk of too much? And how do we control for that? And so One of the things that we're doing is we know that in FOXG1 syndrome, we have too little. One way to address the safety of too much is to conduct safety studies in animals that are healthy animals and have 2 copies. And then when we administer the gene therapy, we actually have more than 100%. Right? So we overshoot, if you will. We push the boundaries in healthy animals that have 2 copies, so we are above 100, and we do the safety studies to assess potential toxicities. Those studies are ongoing, right? We're in the in-life phase right now, and this is how, so safety is front and center. We want to treat our patients, our kids, but we first and foremost want to make sure that we enter with as much safety data as possible. And that is why we're actually taking a more stringent approach using more than 100% expression to make sure that we define well those boundaries that Tim is talking about, which are critical.
So in addition to injecting the virus into wild-type mice to drive the overexpression of FOXG1, we are also doing efficacy tests with the 3 different dosage. So the effective dosage that we saw in the mouse model that I presented today, we are using higher dose and lower dose efficacy test at the moment to find what's the range that is still effective, but it's not toxic. So that study is ongoing. It will be completed early next year.
Hello. I asked this question at the conference 2 years ago, but I wanted to re-ask it for any new parents that are here. My daughter's Rose. She was diagnosed 7 years ago, and she has I was told, severe deletion. And early on, we were told the cures, if there were any in the works, would only work for children with mutations, not deletions. And I wanted you guys to just clarify that for any new parents and maybe the differences that would.
Right. Our therapy will work for the children with the FOXG1 deletion because the cause of the problem in the case of deletion is still same, that there is a reduced level of the FOXG1, and we are delivering the functional FOXG1. So I think that will work the same way. We are testing in the FOXG1 deletion model as well. And the other testing is that we made the FOXG1 model with the missense mutation, and it's a good population of FOXG1 syndrome with the missense mutation. And we tested this drug in missense mutation model as well, and there's a significant restoration at the brain cellular levels in missense mutation as well. So as I said, that we are aiming to cover all range of FOXG1 disorders.
And I think, Sue, when the different types of mutations, they affect the FOXG1 protein, right, in a similar way. So
right. So deletion definitely affect the same way. FOXG1 missense mutation can have different disease mechanism, but when we add functional FOXG1, there's a competition between mutant form versus wild type, normal form of FOXG1, and I think adding normal FOXG1 there has the same level of advantage.
Hello, everyone. This is June from Taiwan. I'm sorry, Terry. There are parents around the world, and I bet everybody wants to know the patients overseas and not in the United States. How can we participate in the clinical trial? How our hospital, clinics, how we work? Do we have to move here, or is there any other ways? Thank you.
Guy, do you want to take that?
Yeah, so I think that the answer is it's still to be determined because we still need to go through evaluation of the clinical trial, both design and geography. We haven't landed, we cannot conduct a clinical trial everywhere. We're going to, but in a way, the clinical trial is not the ultimate goal. The ultimate goal is an approved drug. And a clinical trial is a necessary step in order to get to an approved drug. We want to get to an approved drug as quickly as possible. And this means that part of it will be to drive a clinical trial plan that is as lean and efficient as possible, which by definition will also mean a relatively, as much as possible, small number of patients that is sufficient in order to seek approval if the treatment is successful. So there's a balancing act here. So we may not be able to, there's going to be a limited number of patients in the trial that will help drive a faster approval if successful. I guess that's what I'm trying to say, is that we may not be able to accommodate clinical trials everywhere. What we're trying to do is move as fast as we can.
And Guy, do you want to just explain what we're trying to do with the accelerated approval?
So this is something that we plan for, but ultimately will require regulatory agency, approval. As we plan our trial strategy, what we're trying to do is enable, to the best of our ability, the phase 1/2 to enable a request from FDA and other agencies to get an accelerated approval. In other words, usually the classic, if you will, clinical trial paradigm is you do a phase 1 and you do a phase 2 and then you do a phase 3. And in some, depending on the indication or the disease, sometimes you need to do more than phase 3. In fact, for most indications, you need at least 2 successful phase 3s to get approval. In the case of rare diseases, usually 1 phase 3 if successful is enough. And we, as well as others, are going to push the boundaries and actually try to accomplish an accelerated approval path just based on a phase 1/2. That requires a very careful trial design and, of course, a successful treatment effect. And part of that trial design to enable us to go with an accelerated approval has to do with what Ellie and I will talk about later, where we need your help to learn more about certain symptoms and outcome measures in FOXG1 such that we can set the stage for an accelerated approval. So this is definitely a goal and a desire, but ultimately it'll be up to the regulators to decide.
Yeah, and I think this is a good point because I do get this question. Our goal is not the clinical trial. Our goal is the approved drug and then to focus on access. The clinical trial, like Guy mentioned, will be small by design, and our inclusion-exclusion is going to be picked on what could make the most successful clinical trial. So this is something to really keep in mind. Our goal is full access for all patients. And I think, Terry, do you have a question?
Can you hear me? Okay, cool. I was going to clarify a few things. So one of the things that happens is when you do a toxicology study, which is the precursor to a clinical trial, is you take healthy wild-type rats or non-human primates, monkeys, and you inject them with the gene therapy in the same dose and the same method that you would do in a human being. And the purpose for that is by taking a healthy animal that we know has been engineered to be healthy. We know there are specific issues. Like if they're not just taking an animal off the street, these are engineered to have no problem. Hear me now? Okay. So they're engineered to not have any issues. So if any issue does occur, we know it's from the drug. And I think what the doctor was, trying to alluding to is AAV9, the vector that we're using. This balancing act, and I think that's why the next step, the step that they're in right now, your team is in, has to determine what is the safest dose so you don't harm the children, right? The second thing that Nonasha was mentioning is it's like a sliding scale, right? If you treat a 2-week-old, that child will most likely be towards the cure. If you treat a 15-year-old, it'll be more towards a treatment. I think that should be very clear that, I pray we cure all of your kids. but it's going to be a sliding scale towards from a cure to a treatment. And when you do your clinical trial, your goal is you have very few number of patients. You might have 8, 10, 20 patients, and you have one shot on goal. And what you're doing is, by running these natural history studies, you're basically saying, I know that a patient at 4 years old is able to start walking, and at 8 years old, they stop walking, right? And your goal is to try to find patients that you're able to show that you're able to treat the disease so they continue walking, as an example. So that's where it might be difficult in some scenarios where you might choose those children that you think that should be treated, but the ultimate goal is those 8 will lead to everybody getting treated, not just the 8 that are on there. So the 8 that are selected, or 10 or whatever will be selected very carefully, right? And I think those are the things that are going to be very difficult conversations and very difficult things to don't know. But in the end, it's for the greater good. It's not just for the 8 children, it's for everybody, right? And I think that then the last thing I'm going to say is the path that you guys are going down by doing it yourselves and not relying on someone else's IPO or financials to kill your program. Because I have gotten 40 programs on the pipeline of companies that have dropped programs or they're stuck. You don't want to be in that predicament. Do it yourself, get it to the finish line, and then try to find an ethical company to bring it the rest of the way, right? Someone that cares, that's going to want to take it forward. So I'm just saying, you guys are doing the right things. I just want to clarify that, I pray that, you treat the kids and it becomes a complete cure, but there's going to be a sliding scale there, and I think we should set expectations.
Do we have any other questions in the room? I think we've got one here.
Yeah, so on the disease and science part, I was just curious what the duplicated cases, how that ties in. Our daughter's duplicate, so maybe she has more FOXG1, and what, if anything, you guys have learned or where that's heading in terms of treatment. And
Yeah, so this is a great question, and a question that we are actively engaged with between Sue, Ellie, myself, with ongoing efforts. So when we say that we want to treat the broader FOXG1 patient population independent of the type of mutation, this is our goal. The one case that is more complicated, or could be more complicated, is the case of a gene duplication, where there could be too much. We actually don't know that there is too much. So the fact that there is an extra copy intuitively would suggest that you have more FOXG1 protein. This may or may not be the case. And until we know that it is safe to dose a duplication patient, we won't, because we don't want to do any harm. What we are doing, without getting into the weeds, but we can maybe chat a bit later more, is we're actually in the process of requesting and collecting from families that have duplications, samples, basically blood samples, that then in Sue's lab we're gonna conduct research on to better understand whether or not there is indeed overexpression, what we call, or too much, or lower expression. So the fact that there's an extra copy does not necessarily automatically mean that you make too much. There is a scenario where you could still make too little even if you have an extra. We just don't know. So until we know, we will be careful to not dose with gene therapy to not cause harm, but we want to understand whether duplications express more or less so that we understand whether this current therapy can be open to duplications as well. I hope this answers.
Okay, we have time for one more question. Now, Guy and Sue will be here for lunch, and so you can bombard them with as many questions they don't need to eat. But we have time for one more question. Ryan, are there any Okay.
All right. You have a question. Here you go. Sorry, I have 2 quick questions. The first is, okay. I read that this therapy can grow the corpus callosum in the brain. What if the patient has agenesis of the corpus callosum? Would there be any effect on that? That's my first question. The second is, I saw you injected the pups and the adolescents, but have you injected adult mice? And do you anticipate the treatment to be effective for adults who have FOXG1?
Oh, so agenesis of corpus callosum and whether we can fix it or not is a very good question. And we do have the evidence that the corpus callosum grows more with AAV9 FOXG1 virus. It's not full recovery, but there's improvement of corpus callosum. When we did injection at P1, so neonatal mice as well as adolescent mice. And in terms of adult mouse treatment, we haven't done it. So I cannot tell you how much progress we'll have with the wild type. In general, idea is that the earlier the better, but that should be tested. One thing that I was surprised is that I thought injecting a P30 mice would have much less effect than injection into neonatal mice, but we do see very similar level of recovery. So it's really hard to tell without testing it.
Maybe just to add to the second question, and that follows what Terry said earlier, I think that it's fair to expect, and we won't know until we're in the clinic proper, but to expect a certain sliding scale, right, depending on the age. So I'll say two things about that. The first is, in other neurodevelopmental disorders, preclinical work in mice didn't always predict treatment effect in older patients. In other words, there are concrete examples where treating an older mouse with a potential therapeutic agent resulted with no treatment effect in the mouse. And those kids that are treated as part of a clinical trial now, and this is for Angelman syndrome, are responding to treatment and showing improvement at ages that would not be predicted by the mice. So the models are essential. At the same time, they have limitations. So they can tell us certain things, they can tell us important things, they cannot tell us everything. And they're not accurate predictors of treatment effect in the clinic. So it's important. The experiments that Sue's team is doing are essential, and they help us pave the clinical strategy. But at the same time, we recognize that some answers will be answered in the clinic, and knowing what is the age range of efficacy and what is the nature of the sliding scale of treatment effect in FOXG1 kids, not mice, is only one way of finding out, and that is doing the clinical trial.
Thank you guys so much. Thank you. And